So today the Plant Phenome Journal webinar will be presented by Kelly Robbins and one of his graduate students. Kelly's an assistant professor at Cornell University. He's going to be talking about ImageBreed, an open access plant breeding web database for image-based phenotyping. And his paper was recently accepted and published in the Plant Phenome Journal, which is how we came to know about this exciting work. So Kelly, and then about halfway through, Nicholas will switch over, and then at the end we'll take questions. Given the issue we had with Zoom bombing in the past, I'm going to try and closely monitor things, but if you don't want to see anything you don't want to see, please don't view the cameras, because that's one thing I can't close down. So without any further ado, Kelly?
Okay, great. Thanks for that introduction, and thanks for the invitation to present here today. As Seth mentioned, I'm going to start the presentation. I'm going to spend a few slides just kind of giving a high-level overview of what we're doing with the lab and what were some of the drivers behind this ImageBreed system. Nick Morales is a PhD student in my lab. He'll be taking over. He really has been the driving force behind ImageBreed. A lot of this was his idea. He wrote all of the initial code for the system, so I want to give him a chance to talk about what he put together, because it really is fantastic. Given that we're going to be doing two people presenting, I wanted to go ahead and give the acknowledgments up front. So a lot of what I'm going to present today really has been a collaborative effort between my lab, the Lucas Mueller lab, Mike Gore, and Ed Buckler's lab, as well as the research station staff, where we run a lot of these trials. I'd like to thank our funders, USDA, Iowa Corn, USAID, and the Genomes to Fields initiative has been really crucial for providing a lot of the data that we've used in some of these initial analyses. A lot of people to thank, but I have a few people in bold that I just wanted to highlight real quick. So as I mentioned before, Nick Morales has really been a driving force behind this. Nick Santantonio and Nick Kazmar have been doing a lot of work in terms of collecting data, perfecting a lot of the analyses behind the system. Mallet's a postdoc in my lab who's done a lot of the longitudinal modeling that I'll talk about briefly today. And Sam is a software developer that we recently brought on board, and so he's going to be, moving forward, we have this ImageBreed code base that you'll get a sense of here today, but he's moving forward, going to be taking and maintaining a lot of that code base, since Nick Morales is a grad student, and I want to make sure that he's not spread too thin trying to support a code base like ImageBreed. So just a quick background on my lab for those of you that may not be familiar with me or my lab or what we do. So we're very much on the modeling side. My background is actually in animal breeding and genetics. I transitioned to plant breeding and genetics many years ago when I took a job at a large seed company. But we primarily focus on modeling of both complex systems as well as complex traits. The nice thing about being more on the modeling side is you get to work on a lot of interesting projects, a lot of different, crop species. And so we don't focus on any one crop in particular. But I would say that, within our lab, our research kind of partitions into two different areas. One is around breeding program efficiency. So we do a lot of work with international ag, trying to improve efficiencies of breeding programs, particularly those that have limited resources. And the other is modeling growth curves and genotype by environment interactions, and that's what we'll be talking more about today. So when I switched over from animal breeding to plant breeding, one of the things that I really found fascinating were the genotype by environment interactions. And in animal breeding, it's G by E exists. There are obviously lots of examples of it, but it's really not a major focus of breeding. Moving into plant breeding, it was quite surprising to me just how large some of these G by E interactions could be, and potentially how flexible plants are in terms of adapting and reacting to their environment. So it's always been something that I've been very interested in. And when we think about genotype by environment interactions, obviously this is something that's been studied for over half a century. But in general, you could sort of place these approaches into two maybe high-level categories. So one is what I would consider more the traditional statistical approaches, right? And so this is where we treat environments as kind of a discrete entity, and we treat potential re-ranking of lines or changes in performance across environments as an interaction effect. There's different ways that you can do it. A common way is just to model genetic correlations between environments. Things like the factor analytic model are quite powerful in being able to do this. These approaches are really nice because they're very scalable and they're very accurate for modeling observed G by E's. If you have a big data set and you want to understand G by E interactions in that data set, they're great. They really don't provide much insight into the factors driving G by E, though. On the kind of the other side of the spectrum here would be more of a reaction norm approach, where you treat environment as a continuous covariate, where you could have various environmental indices, descriptors, et cetera, and you treat the performance of a line as a function of the environment. And these models are nice because they provide insights into factors driving G by E. But they don't tend to be very scalable, and they're very difficult to try and implement for end-use traits, right? So if we're dealing with something like yield, which is the culmination of everything that's happening throughout the growing season, but you really just have that one data point at the end of the growing season, it's very difficult to model that as a function of environment. Of course, there are modeling approaches out there, like crop growth models that you can use by collecting a lot of intermediary phenotypes. But again, they're not highly scalable. And so in between, these two approaches, there's this huge gap. And in terms of what we're working on in my lab is trying to operate in between these two areas, right? So looking for ways to be able to actually start modeling crop growth in a very scalable way to allow us to do this across a lot of environments and a lot of genetically diverse material. And really key at doing that is a lot of the advances that have been made in various proximal sensing technologies. So this enables fast and cheap phenotyping throughout the growing season. And so a lot of what my lab is working on is looking at these, phenotypes that are derived from proximal sensing data to try and get a better understanding of the genetic architecture of growth traits, examine the relationship between these traits and key end-use traits. And also develop highly scalable approaches for modeling growth and development and understanding the impact of plant plasticity on genotype by environment interactions in the end-use traits. And so there's definitely a lot of work out there modeling proximal sensing as either covariates or correlated traits with end-of-use traits. We want to move more towards modeling growth as a function and looking at what we might be able to do from a breeding perspective in terms of actually breeding for different growth characteristics throughout the growing season. So in terms of modeling longitudinal phenotypes, this by no means is all inclusive, but more from a statistical background. There are a couple of approaches that are available. So one is more of a multi-trait type of approach, right? Where you would consider phenotypes being collected throughout the growing season as sort of distinct traits. And with this, you can use a covariance matrix to model the covariance between traits being collected at different time points. In this case, we have, here as an example, we have 6 time points, right? And so what you can see is that while these are very powerful approaches, they're not scalable because the number of parameters you have to estimate increases nonlinearly with the number of time points you collect. And so it's good up to an extent. In general, we fit multi-trait models and they work well up till about 4 to 5 traits, and then after that they really become unusable. A different approach to this is to use what we call a random regression model or random regression approach. I think the term random regression is used to describe different ways in which regressions are fit. When we talk about random regression model, what we're talking about specifically is the use of covariance functions, right? And so if we think about a trait that changes over time, right, we can potentially fit a function that models the change in variance and covariance through time as a function of time. And so what you do with these models is you fit a covariance function, right? And so the parameters that you're estimating are really parameters that are associated with, let's say, first, second, and third order of a polynomial type of regression. You're using a spline that can be associated with knot points fit for the spline. But the idea is that the number of parameters you have to estimate is now a function of the order of the polynomial and not the number of time points at which you have phenotypes. So it can be very, very scalable to near continuous collection of phenotypes throughout the growing season. And the way these functions work is, by pre and post multiplying by vectors of covariance at different time points, you can estimate the covariance between any two time points within the space of your phenotypic data. And so these approaches are very, very highly scalable. And so what we've been working on recently is developing these types of models for growth modeling in plant breeding context using phenotypes derived from proximal sensing data, right? And from this, if we were to take this example here, if we were to fit all 6 of these time points, we would have to estimate this large variance covariance matrix here. We can potentially fit a polynomial to model the changes in variance and covariance, and we can easily transform this back into the 6 by 6 matrix. And what we found, with work in maze at least, is that we can actually reconstruct these variance and covariance matrices for a large number of phenotypes, quite accurately using relatively low order polynomial functions and splines. So as I briefly mentioned, when you fit a random regression model like this, there's a couple of ways in which you can go about this. You can use spline functions, you can use orthogonal Legendre polynomials, but the main benefit of fitting a random regression like this with a covariance function is, one, it becomes very scalable with the number of phenotypes, and two is that you can start to partition effects into specifically genetic effects, so we can start to estimate genetic parameters around growth curves. And from what we've seen in data so far, these are actually highly heritable, or at least moderately heritable. But you can also partition out into permanent environments effects. And so a permanent environment effect you could fit at a plot level, and what it does is it measures a consistent kind of environmental effect throughout the growing season and models changes in variance and covariance of that permanent environmental effect. And one of the interesting things that you can start to do with that, and we've started looking at, right, is spatial variation is obviously very important within fields, especially here in New York, where it's hard to get a perfectly level field. By implementing these types of random regression models, now rather than just using spatial information, which is sort of the proximity of different plots to each other within a field, we can start using longitudinal data as well, which is we've measured phenotypes on this plot multiple times throughout the season, so we can look at the change of those permanent environmental effects over time. And with the genetic effects, we can start to look at plasticity. We can start to look at things related to plant reactions to environments, which is something that really has been challenging before, especially if you're dealing with phenotypes that are largely collected at the end of the season. Just to briefly kind of show some, of what we've seen so far in terms of looking at this in maze, we're able to reconstruct these quite accurately. We do see that, at least in some of the applications we've done so far, that we do see that we see distinctly different growth patterns for high and low yielding lines. We've started to look at this in alfalfa as well. And what we can see is that, there are certain growth patterns that, here, towards the end of this, we're seeing, actual biomass at harvest across a couple of different cuttings. the parameters change across cuttings, but we do see that, you know, there are certain growth profiles that are associated with some of these key end of season traits. And so, you know, our results so far have been pretty promising in terms of being able to model parameters that are a function of the order of the covariance function rather than the number of phenotypes. This is a very highly scalable approach, but it's highly scalable assuming that you can get, you know, 2 phenotypes from proximal sensing data in a relatively short period of time. And so, you know, what's really become painfully obvious is that the huge bottleneck now is the image processing. And that's really where ImageBreed and what Nick Morales has done has come into play. And so with that, I will switch over to Nick taking control. So please take it away, Nick.
Okay, thanks, Kelly. So yeah, just a brief title slide about what ImageBreed is. It's a set of image processing, or it's an image processing pipeline that was built into the Breedbase web database system. Breedbase has been a work in progress for many years, starting with the Soul Genomics Network, SGN, and then to Cassava Base, and a number of different databases for plant breeding. And so I was working with Lucas for about 4 years prior to starting my PhD with Kelly about 3 years ago. Okay, so just to give a brief of how we're doing the data collection, we use a DJI M600 drone. It flies a pre-programmed path over the field. I use Lychee for the flight planning. So as it's flying, it collects images. We use this MicaSense 5-band camera, which captures in the blue, green, red edge, and near infrared. So immediately prior to flying, we photographed this calibration panel that comes with the camera, and this panel can be used to further calibrate the images that are captured during the flight. And MicaSense provides some code for doing this, and software knows how to use this calibration. Ultimately, ImageBreed is about this process of assigning plot polygons to the images that are collected, and these plot polygon images are related to the field experiment. So what hybrid or what accession is planted there, and also related to the genetics of, you know, if we have high density genotyping for those hybrids, and also if there are other phenotypes like plant height or grain yield that were collected. So ultimately, that's what ImageBreed is about. There are two different paths you can take to accomplishing this. The first one that we were looking at is if you use an orthomosaic. So an orthomosaic can be built using external software like Pix4D or Agisoft. And so from the captured images, you can create the orthomosaic, which spans the entire field. Or what we're looking at now is to skip the external software and to just use the captures directly. Just to give more detail in that, so for the first avenue you can take, images come off the drone or off the camera. They're piped into some external software for creating the orthomosaic. And then the orthomosaic is uploaded to ImageBreed, where the plot images are assigned. So the benefits are that this is easier to implement, and you get this high quality orthomosaic. These software do a great job. The drawback is that they're expensive. You can't always construct the orthomosaic. If the images are too uniform, it's time consuming, and you lose the repeated measurements. As you're flying, you get repeated images of every plot, but when you use the orthomosaic, you end up with only one. So then the second path is to just use the images directly. So from the camera to upload it to ImageBreed, you get, you have all of the captures that were taken, and then from there you can assign plot boundaries to all of the different captures. We want this to be free and open source, and you get the repeated measurements from all of the different captures. What I've seen though is maybe the phenotypes that we're extracting have more noise, and also this, it can be time consuming also. So just to give an example of, like, if we're going down the first avenue, so this is what an orthomosaic or orthomosaic results look like when they come out of Pix4D or Agisoft. And since I'm using a MicaSense 5 spectrum camera, I have V1, blue, green, red, near infrared, and then red edge. And they're all perfectly superimposable coming out of Pix4D or whatever software you're using. So then I upload that into ImageBreed. I have a little video here. So this is what the main dashboard looks like. And so here you see these are different field experiments, and then related to that field experiment, you have different imaging events, so different times that you've flown. And then up here, these buttons, these are the main controls that we have. So here I'm showing how to upload imagery, uploading the orthomosaic. The first screen there is just some information. I just skipped it for time. Then you select a field trial. So the field trial has to already be, in the system. I didn't go into how to upload a field trial, but that's like Breedbase, Breedbase specific. So here we select a field trial, then it'll show you all of the previously loaded imaging events. So here we're creating a new imaging event, and to do that, you give a name. So here I just say give a name for it. Here the type, if it's high or low resolution, the camera type. So I've only really implemented it for the MicaSense camera, but for standard color cameras shouldn't be a problem. Then the date is very important. So you have to give a date for the imaging event. Then on the next step, here's where you actually specify which avenue you want to take. So are you using orthomosaics, or are you uploading the raw captures? So in this video, I say that no, I'm uploading orthomosaics. And then here's where you actually upload the orthomosaic images. Importantly, you specify this drone run band type. So in order to you know, to keep track of what spectra are being observed here. And so for the blue, I select the image for blue that we saw previously. And I do the same thing for the five different bands. And then that's essentially the whole upload process. So you select the orthomosaic images you want to upload, and you give a band type. So once you've uploaded it, that imaging event will show up on the main dashboard here. And then so you can open up that imaging event you just uploaded. You see the information that you input, like the date and the name, and this dashboard shows you all the it shows you a summary of everything that you've done. So here the only thing we have is the uploaded orthomosaic. And that's what it's showing there. And here, so I'm going to run a standard process to actually assign the plot level boundaries. And so, sorry, it's going so fast, but essentially how this works is you select one of the bands, you run a process over that one band, and then it'll apply the same procedure to the other bands. Since the uploaded orthomosaic, the bands are perfectly superimposable. So here I'm selecting the near infrared band to take through this process. The first step of the process is to do a rotation on the image. So we want the plots to be perfectly vertically oriented here. And so here you can play around with getting the rotation just right. This next step isn't actually important. I was working on ways of thresholding pixel values, but that's not really essential here. Anyway, so this is the core of the tool, is where we actually assign the plot level images to this orthomosaic. At the top is an input for the number of rows and number of columns. So I said 5 rows and 17 columns. Then you have to click on the boundaries of the region of interest. So I'm clicking the top left, top right, bottom left, bottom right. So you can pick any region of interest. Like for maize, or here. So this was an alfalfa experiment, and it wasn't perfectly planted. So what I'm doing here is I'm assigning polygons specifically to this first row first. And so it's kind of a bit of trial and error, and I didn't do a perfect job here, but you see the 17 plots for this single row. And then what you can do is you can copy paste that that template, so that's what I'm showing here, copy paste, and then you click on somewhere where you want the boundaries to continue, right? Like for maze, this copy pasting isn't really necessary since it's like GPS planted, and so you can just click the 4 boundaries and then say, I had 50 columns or 10 rows, and the template will show perfectly, but this gives some more flexibility for kind of irregularly planted trials. And then once you have the boundaries looking good, you say where does plot number 1 start here, and so you can say they're in the top left or the top right, and then you say if the plots are numbered in serpentine fashion or in zigzag, and so that's important for the system to know how to actually assign the plot, the field experiment to those polygons. Once you click assign, it'll draw this where these are the plot names. It's just this can be worked on more, obviously, but just to double check that the plot names are actually being matched up to their polygon. And then, so here it shows you that it's going to apply that same process, the rotation, and the plot assignments to the rest of the bands. I won't get into this. I mean, essentially, I want to expand this eventually, but currently the phenotypes. So once you have those plot images, we want to extract phenotypes, right? And so here it shows you what kind of phenotype are you getting, and so we're looking at what we're calling zonal statistics. So these are like mean pixel value, the majority pixel value, the median pixel value, and so that's what that's showing here, that you can select for these zonal statistics, and then you're done. And so it'll go through the process, and we'll store those images against the field layout. Now the second path is more is exciting because we don't have to rely on like Pix4D and other software. And so to give you an idea of what the raw data looks like, this is the images off of the drone. This is the MicaSense camera. So as many of you probably know, you get these _1, _2, up to _5 for the blue, green, red edge, or near infrared and red edge. And so this is the raw data, and then you also have the panel, the calibration panel. So just also wanted to show how to upload that the raw data. So it's the same process from this dashboard. We go to upload imagery. The same thing as before. We select a field trial, then it'll show you the imaging events that were already there, and then we're uploading a new imaging event. So just giving it a new name for that imaging event. High resolution mica sense, and then select the date for that imaging event. Then on the next step is like we saw before, is where you can diverge on either using orthomosaics or using the raw images. And so you upload a zip file of the raw images. It's currently capped at 2 gigabytes, but you can upload. I mean, often you need more than 2 gigabytes, and this isn't actually a problem. But here you select the capture, the zip file with all the images, and then the zip file with the calibration images. And it will, so just as we saw before, here's the imaging event we just uploaded. And the dashboard will show you the date and all the meta information we uploaded. And it'll show you the images that we currently have in this imaging event. I wanted to run through how the process looks for assigning the polygons onto the raw captures. So it goes through a similar process. We select one of the bands to take through, and then it'll apply the same process to the other bands. The first step is a rotation on the raw captures. So here's these are the captures, and they're positioned relative to their GPS coordinates. So every capture has a GPS coordinate associated to it. And so that's how they're being displayed. And so the first step here is to do a rotation on them. And so this, you can drag here and it'll rotate them. There's some, so it'll show you how you've rotated it. Rotated all of the captures in that first step. Then on the next step here, and sorry, there's like a display problem that I haven't worked yet. So the window is huge, and so I'm going to be scrolling like back and forth a lot. But so here are the rotated captures, and in this, then we click this blue button, and this blue button will start. It starts matching the images against each other, so in a pairwise way. And this matching is it's a feature-based match. It's actually the same thing that like Pix4D does, but in a much simpler way. And so it's finding points that are common to the two images, and then based on the most probable points, it overlays those 2 points that are the same in the images. And so you can kind of watch it produce this rectified image of the field. And once you have that, it's essentially the same as if you're using an orthomosaic. here I wanted to show what those automated points look like. So you have, two images side by side, and so there's code for detecting those points and then matching them together. And so that's how the matching works. And then here, it's just same thing as before. We say the number of rows, number of columns, and then we click on the boundaries of the field. So just directly clicking on that on the screen. And this is kind of trial and error. I want to incorporate like ground control points here, so that you can like reuse kind of templates between imaging events. But anyway, here you're clicking the boundaries of the field. 5 rows, 19 columns. I click that blue button and it'll draw those plot polygons. Here I'm not really worried if they're actually, this is just a demo. And then it's the same thing, you say if it's serpentine or not, and then it'll assign those boundaries. And then finish, and it'll apply it to all the other bands. And again, we're getting zonal statistics out of those plot boundaries. So the kinds of phenotypes that are being extracted are essentially mean. I mean, what we're looking at right now is mean pixel values, so that we can calculate vegetation indices, like NDVI. But once we have those plot level images, and so we can look at, we can look at these future phenotypes that I'm interested in neural networks and whatnot. But currently only the useful phenotype right now is these vegetation indices. Like Kelly was talking about at the beginning, we want to model these phenotypes, and so I implemented using SOMR. So it's an R package, and you can account for the genetic relatedness of the hybrids, and the way it's implemented is using the GRM and two-dimensional splines. And here's what the command looks like for a multi-trait model. T1. So these could be different time points that were imaged, or like NDVI at different time points, and grain yield could be one of them. And so this GU is the genetic relationship matrix, and then this spline 2D is the variant structure for the two-dimensional spline. So Breedbase, Breedbase and can store high-density genotypic data, and so using that high-density data, we can automatically calculate the GRM. It's a bit slow, but that can be sped up. And so ImageBreed has the currently has the G2F genomes to field inbred parent genotypes. So we can calculate the GRM from that data automatically. And this is the last slide I have, showing what an analysis would look like in this system. So from this dashboard, go to the top here and calculate statistics. Okay, calculate statistics. You select a field trial. So here, 2019, this is a genomes to field experiment. So we have the next step here is to select some phenotype, select some trait variable, observation variables that you're interested in looking at in a multi-trait model. So here, selecting grain yield, and then alongside, we have, these are all the extracted zonal statistics that I was talking about before. So I'm gonna select NDVI at 54 days after planting, 62 and 67, and grain yield. So selecting those traits for that field trial, and then selecting the Sommer model that I talked about, and here you can say how you want the GRM to be calculated. So Breedbase, ImageBreed, Breedbase have the genotyping data loaded, and then here in G2F, the parents were genotyped, not the hybrids. So this is really for G2F, like to compute the hybrid genotypes from the parents. I won't really get into how that's working, but in the end, you end up with genetic blobs, and the spatial blobs. So since this was a two-dimensional spline model, so just showing the genetic blobs for the multi-trait model that was fit. And below this are the results for the spatial globs that came out of that model. So you have a spatial glove for every plot in the field, and we can save those. So really, I mean, we can save those the results here. I'm clicking on the analysis page. So this analysis page holds the results from that analysis. So it details the model that was run. This was that Sommer model with the GRM and with the two-dimensional spline. And here it shows the kind of meta information about the analysis and the results. So these are the genetic blobs for grain yield and genetic BLUPs for NDVI at 54 days after planting. And just to finalize here, the spatial BLUPs, they're saved in the same way as the genetic BLUPs. So they're you know, it tells you the model that was run to get these results, and the results show up in the same way, except these are spatial, not the genetic, and so they have the information about the row and column and how they were distributed on the actual field. So you can see this heat map of those spatial effects. So a lot of this was implemented in Breedbase, and it was just natural to expand it, for these image phenotypes. Yeah, so to close, this ImageBreed Breedbase system is available in a Docker, like a Docker. It can be deployed just using Docker, and I'm available. Yeah, I mean, we're available for any questions you guys might have. So thank you.
Well, thank you, Nicholas. I don't know if Kelly, do you have anything you'd want to close out with? And
yeah, I would just add, this is a system that's under very active development right now, and so we certainly appreciate anyone that's willing to take a look at it and provide any feedback to us. We have imagebreed.org, I believe. Nicholas is the website that people can go to check out the demo version that we have up and available for it.
Great. Well, let's open it up for questions. Please, I've opened up the chat, so if anyone wants to put anything in the chat, I'd be happy to read the question. While you're doing that, I guess I jotted down a few questions here. So you sort of showed something that looks sort of like a mosaicing feature, but you didn't really say it's going to exactly replace Pix4D or Agisoft. Is that part of your long-term goal, is that the mosaicing would be completed within this?
Yeah, good question. Yeah, I think, it would be, I think, much easier if it was completed inside of the system, so that you don't have to move data around between programs. And but yeah, like you said, I can't replicate what Pix4D or Agisoft does perfectly, but it is it is possible to create a rectified image that we can work with, and we'll see how good, we'll see the differences in the predictive accuracy coming from the orthomosaic or coming from what we're working on. So.
Okay. One of the other, just related to that, while I'm still, looks like we got a couple questions, but just related to that real quick. when we've looked at some of the commercial software like Hyphen or Progeny right now, they're actually doing most of the analysis not on mosaic images, but on something that they sort of back calculate from individual. And I saw you showed you could do it from individual sort of plots. I don't know if that sort of would get around your, the challenges with mosaicing, but both Pix4D and Agisoft, neither one of them are perfect. I mean, sometimes one can mosaic something that the other one can't. And I don't think there's any ideal solution. So replicating one of those is not necessarily I wouldn't say it's going to be a huge goal.
Yeah, I think long-term we want to move away from the orthomosaic step altogether, just because it is kind of, computationally costly, and it doesn't necessarily always provide the most desired outcome. So yeah, we're still working on some of the features. One of the real key things that we wanted to bring with this tool is that, as part of doing this in the system, and because it's built off of Breedbase, which is a fairly popular and widely distributed breeding management system, is that, once you've gone through this process, now you have all of these, derived phenotypes together with all of the trial meta information, pedigree information, genomic information, et cetera. And so it makes, the ability to do a lot of the downstream kind of comprehensive analyses that at least in our lab, we're very interested in, much easier to do. And so, yeah, moving forward, we have a software developer working on this now along with Nick. And so the idea is to phase in and enhance those, processing capabilities as much as we can, as fast as we can.
Okay. Number of questions here from Dr. Tyre Weisner-Hanks. What crops have you tried the plot boundary detection on? Does it work better or worse on given crops?
Oh yeah, yeah. Hey, Tyre. So I've tried it. I've only tried it on alfalfa and maize, and so I think you're talking about the matching step. So like the automated matching, because the boundary, like the plots are defined by the user clicking. So the automated point matching, it works well with alfalfa and maize. And really you only need 1 point to be, you need a single point that is certainly the same between images, and the algorithms that people have. I'm just using algorithms that other people have done for this, and they can definitely detect at least 1 point that's common.
Okay. Dr. Larry York asks, have you tried Open Drone Map for orthomosaics? You also mentioned OpenCV. Is there an implementation for generating orthomosaics in OpenCV that you are aware of?
I've tried Pix4D and Agisoft, or I haven't tried Open Drone Map, and there's not there. So there is an I do know of a OpenCV implementation for it, but it doesn't scale well. So if you have like more than like 20 captures, or because it scales like cubically, so that if you have 1,000 captures, I only got it to work for imaging events where I had less than like 20 captures, but I can share that with you if you want.
Okay. Dr. Jeremy Edwards asks, is there a plan to include 3D features like point clouds to measure traits such as plant height?
Yeah, so we're putting together a project proposal working with some people, some computer vision experts to try and build some of that functionality in. It's not going to be, I would say it's not going to be included in the system anytime in the very near future, but that's definitely on our radar.
Okay. Dr. Jin Zhang asks, how well will this sticking images work when data are acquired in a non-flat area? I'm afraid the stitching process may introduce significant scale issues when images are acquired on a hilly environment.
Yeah, that is maybe a concern, but the drone actually keeps a fixed altitude above ground level, so or at least it tries to. So if it has information about the ground elevation where you are, it will adjust its height, its absolute height to keep a certain ground altitude. So yeah, that should hopefully mitigate any scale problems.
And the last question we have, Tier says, thanks, Nick. And then the last question we have, second to last question we have from Dr. Jenna Jung again. Can we reuse plot boundaries generated from one flight to another flight over the same area later?
Yeah, this is still, I mean, it's definitely possible, especially if you have like ground control points that you can see. So I'm working on a feature to where you can paste previously used boundaries from previous flights or imaging events that you had.
Okay. And what I believe is the last question from Dr. Francisco Nunez Arreola. Is it in your plans to work with remote sensing imagery from doves, in quotes and in all capital letters? I think those are the satellites, right? Yes.
I'm not sure what doves is, but
So I think they're like the small sats, the satellites that, you know, there's, I don't know, a network of 250 of them, or there's going to be, and so it's more on or cubes, yeah, more on a landscape scale than on a, you know, drone scale. So
okay.
the question was just if you had a plan to work with those, but
We don't have a plan right now. I mean, I think ultimately we would want to, expand to deal with multiple, multiple platforms for collecting sensor data. I would say probably on our list before that would be some of the ground-based rovers that, at Cornell they've started actively collecting data from.
okay. All right, well, with that, I don't see any more questions, and we are a little bit over time. So thank you both, Nicholas and Kelly. A lot of people thanking you online in the various chats and whatnot. So great job, and we'll look forward to seeing what you guys do with this in the future.
Thanks for having us.
Thanks a lot.